Welcome to everybody who is online already. You are all muted at the moment, so if you can just maintain that please throughout the webinar. Your video is also set to automatically be off. It's fine if you've turned it on. It's entirely your choice if you want to activate your video, but it's not mandatory. So welcome back. This is our webinar 2 of our bite-sized research series. Today we've got a special guest, which is Dr. Darrell Green, who's going to be presenting to you a presentation on understanding DNA and RNA in bone cancer research. And two of my colleagues, Dr. Zoe Davison and Dr. Vicky Vinader, are also on the webinar as well, so they may ask questions of Dr. Green towards the end, and they're also on hand should other people have questions for them. So just to introduce myself, for those of you that weren't here last time, I'm Matt Coltell-Shaw. I'm the Head of Fundraising and Communications at the Bone Cancer Research Trust. And the reason for doing these webinars is it came out of the fact that we are unable, due to social distancing, to hold our annual bone cancer conference as planned in London next month. So we thought it would be great to try and bring some of the research into your own homes, basically, and to give you direct access to ask questions of our experts, just the way that you would be able to do at our conference. So I think we've got nearly everyone on at the moment. I'd just like to remind everyone that if you do have questions throughout the webinar, please use the chat function. I would try and encourage you not to get into conversation on the chat function, but just to post questions, because it makes it really hard for me, the host, because my job is to try and pick out your questions and make sure we're sticking to time, and make sure that your questions go to Darrell at the end of his presentation. So if you can do that. If you do have any questions related to personal treatment or personal circumstances, then I would encourage you to direct that separately to our support and information team. If you do need to get in touch with us, you can email us back to the email link, the email address, sorry, where the email link was sent to you for this webinar. So without further ado, I'm going to hand over to Darrell now. I'm just going to unmute you, Darrell. There you go. If you want to introduce yourself and then tell me when you want your slides, and I'll...
Well, you can bring them up now if you want.
Okay. There you go. Everybody should now be able to see the presentation slides.
Cool. So, first of all, I should probably say that I'm sorry if you've seen this talk before, because I did give this talk at the conference last year, but apparently it went down really well. So I'm going to pretty much give the same talk again, except I have updated it a little bit, especially towards the end, regarding latest research coming out of our lab. Yeah, let's crack on. So one of the first things that you might do as a patient or a family member, or if you're interested in cancer or bone cancer, you're probably going to go home and Google bone cancer, and you're going to be facing a lot of these new terms that you might not have seen before, like sarcoma, osteogenic. What is a tumour exactly? So really this talk is to really go back to the absolute basics, break it down and then build it back up towards bone cancer specifically, in a way that you might find it easier to understand. So going way back to the beginning. Next slide please, Matt. I'm going to turn into Brian Cox just for the next few slides, because if we're going to understand cancer better and treat it better, then in my opinion we have to go right back to the beginning. And the cancer story that I'm going to tell starts way back, to the beginning of the universe almost. So our sun is a giant nuclear fusion reactor, and pretty much all it does is combine hydrogen atoms that you can see at the bottom there to create helium. So this creates a new atom, a new element, and by crushing those two hydrogen atoms together, you get a release of energy like heat and light. However, when our sun runs out of hydrogen atoms, on the next slide, you'll see that because our sun is of a certain size, it will be able to then combine the helium atoms to produce larger elements, larger atoms like carbon and oxygen. Next slide, please, Matt. However, our sun is huge, but compared to other stars, it's not that big. It's not going to have enough gravity to then crush oxygen and carbon together to create new atoms. So at the end of that second period of its life, it's going to explode and it spits all of these carbon and oxygen atoms out into the universe. Next slide, please, Matt. There are, however, much larger stars that are capable of crushing together these carbon and oxygens, and that's where you get the production of these larger elements, larger atoms like nitrogen, calcium, and phosphorus. Next slide, please. So, had this been a live talk, I would have then asked everybody what was so interesting about those previous molecules that I just had listed. And it is that you are quite literally made of stardust. So all of the elements that are in your body now were at one point created in a dying star. Next slide, please, Matt. So all of these new elements, have been created. They came together on our early Earth. So here's a graphic of what Earth may have looked like around 4 million years ago when it first formed. All of these atoms and elements that were created by dying stars end up combining, and over time they form DNA and RNA. Now you give that a bit more time and the DNA and RNA then evolves even further, and it becomes part of cells, these single molecule beings that exist. And then given a bit more time and the right conditions, these cells then develop into multicellular life. So you can see how stardust quite literally forms living beings. Next slide, please. So to understand cancer, we have to go back and work our way backwards. So from multicellular life, which is where cancer arises, we then take a step back and we look at, okay, what makes us, what makes cancer? And it's cancer is an abnormality of cells. So we're going to go over what cells are, so the basic units of life. And in an average person, this was calculated to be around 37 trillion human cells make up an average person. And then within those 37 trillion human cells, there are 100 trillion microbes in your gut and on your skin. So it could be worth saying that you're more microbe than you are human. Next slide, please. So here is a pretty standard model of a cell, and within your cell, you're going to have the DNA. This is like the textbook instructions for that cell and for you. You have RNA, which is produced by reading the DNA. So these are the copy notes of instructions that are needed at the time. So the DNA will contain the entire manual of how to create a multicellular being, but not all of the DNA is needed all at once. So the sections of DNA that are needed at certain points, a copy will be taken in the form of RNA, and then the RNA is translated into protein, and protein is the action molecule that carries out the actual work of the DNA. So a lot of these actions will be like to determine your mood, to tell a cell to divide, to tell a cell to go through programmed cell death, or they'll act as hormones. Next slide, please. So cells come in all shapes and sizes depending on the role that they need to perform. So you'll see an immune cell here has these arms and legs so that it can reach out and capture viruses and bacteria to get rid of them. You've got skin cells that are very flat to cover your entire body. Brain cells are going to have all these arms and legs as well so that they can connect to other brain cells. And there's a microscope image of a bone cell there where it sits within the bone and it secretes this matrix that forms up the bone. Next slide, please. So cells must divide into new cells when they're needed. So most bones have only got a finite life expectancy. But you are born with a very special population of cells called stem cells. These are present throughout your body. You're born with them and they stay with you for the rest of your life. And they don't have that time limiting function. So these cells are a lot rarer, but they produce new daughter cells to carry out the functions of your body. So within your bone, you're going to have stem cells. They don't instantly become bone cells. They have to go through a stage of maturation, almost like a teenager becoming an adult, that we would call a precursor cell. And for every organ, you would have a specific stem cell that produces that organ cell. So as I said, in the bone, you have stem cells that produce bone cells, and in the kidney, you will have kidney stem cells that produce mature kidney cells. Next slide, please, Matt. So in the bone, this happens in very specific regions of the bone. It doesn't happen in the entire bone. And these regions are called the growth plates. So the image below is of a femur, and the lines in red are what we would call the growth plates. So this is where most of the cell division takes place. This is where most of the bone stem cells reside to produce bone cells. And this is switched on to become a lot more rapid during adolescence, when your skeleton is obviously expanding in size. So in bone, this also happens to be where most bone cancers arise. So at any stage along that pathway between the bone stem cell, precursor cells and bone cell, a cell can receive damage, become cancerous, but it typically always happens in these regions of fast cell division. Next slide, please. So what exactly do we mean by cancer? Because you do hear of tumours, you hear these words benign and malignant. So cancer, if you take an abnormal cell that has received damage in the bone growth plate, it will then start to divide abnormally. So you get an overproduction of cells. This overproduction of cells will then form a tumour. And tumours can be benign or they can be malignant. Now, malignant is the cancer side of it. And what causes, or what is a key feature of malignancy and of cancer, is its ability to spread. So if you have a tumour that doesn't have the capability to spread, it's what we would call a benign tumour. And these are mostly not life-threatening. They can be removed with surgery, and that's the end of the story. But if you have abnormal cells that have a capability to spread, then this is what we would call malignancy or cancer. These cells, for several reasons, find their way to a local blood vessel. They travel in the blood and they produce these secondary tumours. But this is a very, very complex process that tumours and cells have to go through. Next slide, please. So what causes cells to be abnormal? If we go even further back in our timeline, then we will see that DNA and RNA is the brain of the cells, or you can consider it to be the brain. So if we go forward a couple of slides, so there's the DNA is the brain of cells. Now we're getting to the real cause of cancer. So DNA is supercoiled into chromosomes because in every single cell in your body, there is 2 meters worth of DNA. So obviously to fit that 2 meters worth of DNA into a microscopic cell, it has to be super coiled and super wound up to cram inside the cell nucleus. So on the image on the left, you'll see a typical DNA molecule that you've probably seen many, many times. This gets super coiled and wound up into chromosomes, and this is how your DNA is stored. And on the right-hand side of the image, you'll see what we call a karyotype. So this is a typical human chromosomal picture under the microscope. And obviously you have one chromosome from mum and one chromosome from dad. So together you have a pair of chromosomes that make you. And then the last two chromosomes are the sex chromosomes, and that determines your sex. So you're X and Y if you're a male, and you have two X's if you're a female. Now, every time your cell divides, it has to make an exact copy of that DNA, that whole 2 meters worth of DNA sequence. It has to make an exact copy. Next slide, please, Matt. So over your lifetime, DNA will accumulate changes, or what we call mutations. And this will happen just purely from being alive. There was a figure that once came out that your DNA is permanently damaged every 8 seconds, just purely from your cell performing its job, turning over. You'll get background radiation coming from space. There'll be environmental toxins that you're exposed to. And it's been worked out that about 6 or 7 mutations in 350 genes that are specific to cancer out of your total 20,000 genes is enough to cause cancer. So the sequence examples that I've got on the bottom left, you have an example of the normal sequence, and then we have 3 examples of what a mutation might look like. So on the first faulty line, we'll see that rather than saying ATC, that T has now changed into a C. So that would be what we call a mutation. You can also have mutations where an extra letter has been added where it shouldn't have been. So on the second faulty example, you see a T has been whacked on the end there where it shouldn't be. And you can also have what we call deletions. And this is where one or a few sequences are completely removed from a normal sequence. And these are types of mutations that can cause cancer. Next slide, please. So if I've just said that mutations accumulate over your lifetime, why would we see cancer in children? We know that the majority of cancers are diagnosed in older people because it's taken their entire lives for these mutations to happen. So the obvious question is, as I said, why would we see cancer in children? Why me, why my sibling, and why my child? Next slide, please. Now, childhood cancers, and especially bone cancers, are very different to adult cancers. So adult cancers are very busy. They will be full of different types of mutation. But when we look at bone cancer genomes, they are what we would consider to be fairly quiet. Because these cancers develop in younger people, they haven't lived long enough for these specific mutations to occur. So the genome is fairly quiet. But what we see are, rather than these small changes that are spread across the genome, we get these huge chunks of DNA regions are cut out or missing. So for example, with osteosarcoma, there's a very important gene called TP53 on chromosome 17. And there's a huge region of this chromosome that is missing that we think causes osteosarcoma. So in the previous slide, I showed you some sequences and where most mutations are one change. Well, the bit that's missing out of TP53 is usually around 10,000 of those letters are cut out and missed. And this causes the rest of the genome to go haywire, basically, because TP53 is a very important gene in protecting against mutations. So on the left-hand side, you'll see a normal human karyotype with the TP53 gene highlighted there on chromosome 17. And then when an osteosarcoma tumor has evolved enough, because it doesn't have that TP53 anymore, the whole genome goes crazy. So on the right-hand side, you have a karyotype from osteosarcoma, where you have multiple copies of individual chromosomes, you have bits missing, you have bits rearranged, and this is what makes the tumor so aggressive. On the next slide, we'll see that it's a similar story for Ewing sarcoma, where it's a fairly quiet genome, but a region on chromosome 22 that contains the EWSR1 gene is cut out, and it's moved and it's pasted into chromosome 11 next to a gene called FLE1. Now, EWSR1 and FLE1 on their own are normal genes that we need for our normal turnover. But when they're combined in this way, it forms a new gene that makes the cell divide rapidly. On the next slide, we're going to see that chondrosarcoma is a little bit different because it behaves more similarly to what we'd expect in adult cases, where you get these single changes. But as research has been done over the last couple of years, we are starting to see that there are these chromosomal regions involved as well. So chondrosarcoma is a bit of a mix of what we would consider an adult cancer and a childhood cancer in the way that it's been mutated. Next slide, please, Matt. So really, our best guess is that the sped up cell division within the growth plates causes the cells to make a huge mistake when making copies of their DNA during the DNA replication process. And this is what we think likely causes bone cancer. It is nothing more than a sporadic accident. And the way that I like to think of it is if you remember the textbook example that I used about DNA, if you imagine being given a really thick textbook for homework, and you're told to copy every single letter, every single word out of that textbook as part of your homework, but your deadline to hand that in is the very next day, within 24 hours. You're very likely going to make a mistake at some point, versus slow cell division, which is where that person could have been told, okay, your homework is to completely copy this book, but you've got a month to do it. So because of that sped up cell division, that sped up necessity to have to make an exact copy of the DNA, a huge mistake is made, and that's what causes cancer, or bone cancer in this case. Next slide, please, Matt. So I'm now going to talk a little bit about research in my lab and what I do. So I am an RNA biologist. I work on RNA. So if you remember, these are the copy notes of instructions needed at the time. Now I work on RNA for several reasons, but something really interesting about RNA is that if an RNA molecule is present in the cell, then you can bet your bottom dollar that the cell needed that gene transcript at that time. So it gives us something a little bit easier to work with when we're looking for drivers of cancer, because if you look at the DNA, the whole DNA molecule is there all the time. So it's very difficult to pinpoint exactly what is driving your specific tumour if there aren't markers to show what part of the DNA is active at one time. With the RNA, we know that the RNA is only there if it was needed at that exact time. Next slide, please, Matt. So I am an RNA biologist, but my interest is in applying RNA biology to metastasis, because this is the real difficult piece of work with cancer. So metastasis is the clinical term for what we mean by when a cancer spreads. And I work on circulating tumour cells. So these are tumour cells that are very different to the entire primary tumour. They've somehow managed to be different from a tumour cell where its role in life is to divide and divide and divide. A circulating tumour cell, or a CTC, has some ability that makes it leave the primary tumour, it survives the circulation in the blood, and then it produces secondary tumours. And as I mentioned earlier, this is an extremely complex process. And these CTCs obviously have to be very different to tumour cells in order to carry out this process. So next slide, please, Matt. So over the last 3 years, I've been working on circulating tumour cells. We know that there is one circulating tumour cell per billion blood cells in an average cancer patient. So as you can imagine, it has been extremely difficult to isolate circulating tumour cells from patient blood. But we're working with a very good company who developed a microfluidic device, who helped us develop a new method to find circulating tumour cells in the blood. And under the microscope, they look extremely weird. They're nothing like we've ever seen before when you look at normal cancer cells. And on this slide here, this is what steps we took to prove that we were working on circulating tumour cells. We hadn't just found something random. So we looked at the bright field image. So this is just looking at the cells down the microscope. As I said, they look like nothing we've ever seen before. They look very strange. We apply a Hurst stain. So this stains the live DNA. So we know that these cells are actively alive and they have DNA inside them. We then stain with a well-known sarcoma marker. So we know that we're dealing with a cancer cell that, even though we found it in the blood, it did come from the primary tumour. It wasn't something random in the blood. We have to prove that this wasn't an immune cell that we've picked up by accident. So we use another stain called CD45. We put that on, and if we had accidentally picked up an immune cell, that would have come up green. But we see that it's completely dark, so we're pretty confident that so far we have a sarcoma marker. So then we merge the images, we see our Hurst, we see our sarcoma marker, and we also carried out some sequencing, and we see key genes that are involved in cancer that you wouldn't find in the blood. So we're pretty sure that these are circulating tumour cells. Next slide, please, Matt. So another really cool thing about RNA is that you can reprogram RNA. You can destroy unwanted RNA molecules by genetic engineering, and you can create synthetic RNA molecules. So if we see an RNA is missing, say it has a protective effect against cancer, we can try and mimic the effect of that RNA being there and replacing it. So in the top image, we've added fluorescent probes to a specific RNA fragment that we have synthetically engineered, and then we test different concentrations of that RNA with the fluorescent probe so that we know that the cell has taken up that RNA. So you trick the cell into uptaking that RNA where it will then have an effect. So we carry out those experiments, we find out how much RNA we need to enter cells before it has an effect, and then we create a synthetic metastasis model. So the bottom two figures there, this is where we lay cancer cells along a plate, and we quite literally cut or slice something down the middle, and that creates a gap. So you now have cancer cells on either side of that gap, and we use this to model metastasis or the cancer spread. So over time, we use a video camera down the microscope, we leave it for 24 hours, and then we can see how the cancer cells close that gap, and that is our model of metastasis. So the control section, this is where we've just put cancer cells on the bottom of the plate, we've let it to grow as it naturally would, and you can see that within 12 hours it's fully metastasized and it's fully closed that gap. But there are certain RNAs within those cancer cells that if we switch them off using reprogramming methods, you completely slow down metastasis. So in the RNAi boxes there, you'll see that at 12 hours, this gap hasn't closed. These cancer cells haven't managed to metastasize fully and close this gap. And even at 24 hours, they still haven't entirely closed that gap. And we do loads of statistics on this, and we prove that it's a significant effect. This isn't something that's just happened by chance. Next slide, please, Matt. When we do this in whole tumours in the lab, this is just another example. So the previous slide was in osteosarcoma. This slide is in chondrosarcoma. So we can see that for different bone cancers, we can create the same effect by reprogramming the RNA within bone cancer cells. So on the left-hand side, you'll see an untreated tumour. So all of the blue dots are the Hurst stain. So that shows that the DNA is alive and well. And then on the right-hand side, you see that the tumour is completely dissipated. And you have these 3 random cancer cells that don't know what to do with themselves anymore. And if we let that experiment run even longer, then they end up dying. So, but it's all well and good doing that in the lab, and it's all well and good doing that in cells. But what you really need to do to prove that something is worthy of a new treatment is to then carry out these experiments in live animals. And with Ali Gartland in Sheffield, who's speaking next week, if it hadn't been for the COVID-19 lockdown, we were then about to start experiments to show this work in mice in chondrosarcoma. Next slide, please, Matt. However, for the purposes of this talk, I and others have some research coming out any day now. This was accepted by the journal. It's going to be published, so nobody else has ever seen this data that I'm going to present in the next few slides. And this is in osteosarcoma. So just as a bit of background before I show you the actual data, you may remember on one of the previous slides I showed this image of a tumour that are the coloured circles in the middle, but what I didn't mention were the white circles on the outside and the arrows. When you look at cancer, you can't look at the tumour by itself. You also have to look at what we call the tumour stroma. And these are normal cells, these are non-cancerous cells that tumour cells hijack and recruit locally to support the tumour cancer growth. So if we were to magnify in on that tumour stroma, on the right-hand image, you'll see a zoomed-in image of what we would call the tumour microenvironment. So when you deal with cancer, we're now starting to realise that you have to deal with more than just the tumour, it's the tumour microenvironment as well. I have two examples there of a cell that would form the tumour stroma, and these are a particular cell from the immune system. Now, we know that there is an immune cell called an M1 macrophage, and this is typically anti-tumour. These prevent tumours from growing. They try to kill cancer cells off. But we also have what we'd call an M2 macrophage. So this is, for some reason, an immune cell that becomes pro-tumour, and it helps the development of the tumour, and it helps the tumour to spread. And on the next slide, we'll see that Dominic Heyman, who you may have seen on the BCRT website, as BCRT funded one of his projects a couple of years ago, he found that patients without metastasis, if you were to examine their tumour microenvironment, they had many more of these M1 macrophages or anti-tumour macrophages, immune cells in the tumour microenvironment. But when he looked at patients with metastasis, when you look at the tumour microenvironment, there were a lot more of these M2 pro-tumour macrophages. And this was a really key finding between patients without metastasis and patients with metastasis, because we know that once bone cancer metastasizes, it then becomes a much larger problem to deal with. So that's the background of the osteosarcoma work that I'm now about to show you. So on the next slide, with Dr. Katie Finnegan, who you may remember also gave a talk at the conference last year. She's based in Manchester. We performed a lot of RNA sequencing on whole tumours and on blood samples from patients that we collected in Birmingham. We found very specific RNA molecules that we suspected had a role in promoting bone cancer metastasis. So using some of the genetic engineering that I mentioned earlier, where you can create synthetic versions or you can delete RNAs that you don't want, we then took this through cells and then we did it in live animals to show that it can work in a living being. So on the control panel, on the left-hand side, the top left-hand side, we've injected human cancer cells into a mouse, and then we've allowed the tumour to grow. The tumour grows fairly quickly, and by day 49, that mouse has now been killed. And if we examine the mouse lung, you can see that that cancer metastasized very quickly. On the bottom images, alongside those, you will see where we've silenced a particular RNA molecule that we found in clinical samples, so in patient samples, patients who donated samples to research. We found that RNA molecule, and then in the mice, we engineered that RNA molecule to not exist anymore. And what you can see is that within the same time frame, so 21 days, 28 days, 35 days, that tumour is really struggling to get going. And day 49, the mouse is still alive. The tumour has got bigger, but importantly, if we scan the lung at the same time, we can't see any metastasis. Now, when we remove these tumours from the mice to analyse them, to find out what's going on the right-hand images, the top right, you'll see under the primary tumour, the WT, this is the wild type, so this is where we've just left the cancer to grow as it would. The RNA molecule is in red. The green molecule are these M2 macrophages that promote this pro-tumour. And when you look at the silenced RNA version, there's barely anything at all. And then on the bottom right images, you'll see that we see the same thing. When you remove the tumours from the lung metastasis, it's the red is the RNA that you see in the wild type that you don't see in the silenced the lung when we look at the lung. So that's work that we've done with Katie Finnegan, and she's got some crazy good research coming out over the next year or so, where she's looked at this pathway in other cancers as well. Next slide, please. So almost at the end, so I just want to say that what I was talking about earlier, where you get this evolution of DNA in cells and multicellular life, there is a potentially really cool thing that was starting to gain traction, which was quantum biology. I'm really fascinated to see if this kicks off, because this could be the future of cancer research, because this is taking things back to an atomic level to see whether you can modulate cancer by modulating the actual atoms that create the cancer. There's a great documentary on BBC iPlayer, or at least there was, by Jim Al-Khalili, who used several examples in biology of how quantum biology was answering old questions in biology, and I really think that was fascinating, and I really suggest it if you've got time. On the next slide, just a quick note to say thank you to the organisations that fund me. Obviously this wouldn't have happened, none of this research would have happened without public donation. I don't receive any government money at all, so I just want to thank these charities and thank you for all of your efforts with the fundraising and the donations, and I hope that the figures that I've shown you today really show you where your money goes. When you donate, we really are making great strides forward. So thank you.
And there we go. Thank you ever so much, Darrell. That was really, really good. Really, really good. Your research, the latest results that you've just shared with everyone really seem promising. Really good.
Yeah, I'm really excited about it.
Yeah, really good, really good. I think actually I'm just gonna, I know some questions have come in, so I'm just gonna actually unmute Zoe, because I think Zoe's got a question for you. So Zoe, if you don't mind just asking your question whilst I go to the questions on the chat.
Yeah. Thanks very much, Darrell, that was great. I was just going to ask about the RNA engineering. Is that something that you see as just being a laboratory tool, or do you think it could make its way to clinic?
So for a long time, there were some really good results happening in the laboratory, but one of the big problems that pharma had was how you deliver these RNA molecules to the cell that you need them to go to. So they worked fantastically well, but delivery was the problem. So over the last few years, research has really been focused on not necessarily the mechanism of how the RNA is working, but how to deliver it to the specific body area. There have been some really interesting moves forward with this. So I don't know if you've heard of the genetic disorder SMA, spinal muscular atrophy, but there is a drug that came out, or NICE approved a drug recently, that it is an RNA oligo drug. And they've modified some of the RNA. So similar to how I added fluorescent probes to the end of my RNA, they've developed obviously a secret way to protect the RNA and to make sure that it gets to the cell that you need it to get to. There are some cardiovascular diseases, some liver diseases where pharma have developed ways to get RNA to the heart and to the liver. For cancer cells, things are going to be a little bit more tricky, but I know that there are several clinical trials that companies think they've cracked it. So I think in the next few years, RNA therapy, it is on the horizon, and it will be a game changer, I think.
Excellent.
There is a question that's come in. I'm sorry, I'm not sure of the name because the tag is Hawaii. But when did TP53 testing start? I was tested, but I noticed that a lot of people don't get tested or even know about it.
So what was that? When did it?
When did the TP53 testing start? This
So,
individual tested, but they noticed that a lot of people didn't get tested or even know about it.
So I don't think it's a routine thing to have bone cancers genotyped. This is mostly research samples that we've managed to generate to show that TP53 is the predominant driver of osteosarcoma. So TP53 is responsible for most cases of osteosarcoma. There is another gene called RB1 or retinoblastoma 1 that behaves very similar to TP53. But again, it's that same huge deletion that drives that tumour. For a clinical input, I don't think TP53 really is something that would lead to a different treatment per se. One of the problems with bone cancer treatment and these targeted therapies, they're doable in adult cancer. So if you think of breast cancer, there are about 15 to 20 different types of breast cancer based on the genotype or the mutations that have taken place. And we know in breast cancer that certain mutations respond differently to certain drugs. But if you remember, we're talking about single base changes, single letter changes within the DNA that those drugs can target. Bone cancer is going to have to be treated very differently because they're not these single base changes. They're huge chunks of DNA that are missing. So it's very difficult for those similar sorts of drugs to work the same in bone cancer as they do for other adult type of cancers.
Amin has got a question that is the microenvironment affected by inflammation caused by the M2 macrophage and other immunogenic agents?
Yeah, absolutely. So we know that inflammation there's a relationship between M2 macrophages and inflammation, whether the M2 macrophages are driving inflammation or whether inflammation is the signal from the tumour to recruit these M2 macrophages. So these M2 macrophages, they start off life as an innocent immune cell. It's the tumour itself is reprogramming these innocent immune cells to become M2-like and protective. We know that inflammation is involved. We know that inflammation does make it worse, but we're not entirely sure what comes first. Is it inflammation that drives the M2, or is it the M2 that drives the inflammation? But it's certainly a key part of cancer research. There's huge amounts of research looking at inflammation alone as a driver of cancer.
Nick has asked, something like half of chondrosarcoma sufferers have no obvious mutations in the usually suspect oncogenes. What should be done to improve the chances of finding the driver mutations?
What are the chances of finding them, did you say?
What should be done, I think it's done as a type, what should be done to improve the chances of finding the driver mutations?
That really is just going to be numbers, patient numbers. The more samples we receive through research, then the more chance we are going to have, not of just finding mutations, but of proving that they're linked to the cancer. So, you know, it's one thing having a couple of chondrosarcoma tumours, say, and then spotting mutations between those couple, because everybody is so genetically different. It's very difficult to pinpoint what is driving that cancer. How do we know that that change isn't just specific to you? So, as I said, over your lifetime, your DNA accumulates mutations and changes, but not all of them are bad. Not all of them are going to create disease. They will become part of you. So if you only have a handful of patients, it's difficult to say whether a mutation is just because that's you, or is it driving the cancer. The more samples you have, the more people are recruited to trials and to things like Iconic. We can then do stats and other mathematics and things like that, that if we see a specific mutation or a specific change keeps recurring within the same place across all of those different people, then we can pinpoint a lot more easier that mutation has a role to play in chondrosarcoma. So yeah, the answer to that question is we just need samples. If you do have bone cancer and you are approached to take part in a trial, then every sample counts. Because it's such a rare disease, we are constantly facing an uphill battle. So if you are a patient and you have donated, I can only say thank you, not enough times, because because we're dealing with such small numbers of patients, it really is making a change. It really is making a difference, and it makes it easier for us to spot these mutations.
Nick's just done a response. So, okay, does that mean the driver mutations are in the WT, or are we looking in the wrong places altogether?
What would he mean by the WT?
I'm not sure. Nick, if you want to quickly type on wild type.
Wild type. So the wild type is what we would consider to be a cancer that we haven't done anything to it.
Okay, so he's asking, does that mean that the driver mutations are in the wild type, or are we looking in...
Yeah, sorry, yeah. So you would have to look at the wild types, but it's difficult to find or to say that something is a driver mutation in one person or just a few people. You would need to see that same mutation in many different patients with the appropriate statistics and maths applied to it to say, okay, that mutation is a driver mutation. Now that would have to be validated because there are many association studies. I had it in my original talk, but I took it out, that when I said bone cancers are likely a sporadic accident, there are trials such as the 100,000 Genomes Project that they say that they might find that some people are predisposed to making those sporadic accidents. But I'm a tad weary of those studies because they're association studies. If you look at enough data sets, if you look at enough genomes, we're talking tens of thousands, then of course you're going to find something. You can correlate anything with anything if you look at enough samples. So there has to be a sweet point where you have enough samples that you can say, yes, that is a driver mutation in the wild type, but you then have to validate that in the lab. So you could take normal bone cells, for instance. You can create that same mutation that you saw in a patient, and then if it turns into a tumour in the lab, then you've pretty much hit the nail on the head that is a driver mutation then. So everything has to be followed up.
Nick's responded again, just saying thanks, and another message, but I'll make sure you see that afterwards. There's a question from Chris Hassel. Is it possible to detect problems like TP53 in childhood, early years, as a predictor of a propensity towards osteosarcoma or other cancers?
So there are some heritable genetic disorders that are linked to P53. So some families do have TP53 mutations that are passed on down the generations, but this becomes known very quickly. Like if you had these disorders, you would know about it. The trouble is with bone cancer, it's such a, it comes from nowhere. There's no real clue that it's going to happen. So you could argue, for argument's sake, you could say, okay, well, should we perform TP53 sequencing on every child? Maybe you would catch a TP53 mutation there. We're not entirely sure how long it takes, from that initial TP53 abnormality, how long does it take to go from that abnormality to tumour development? We think that bone cancer happens very quickly. So, would there be enough time to screen for that TP53 mutation, to get the results, to have all the analysis done by the time you notice that tumour's there? I don't know. I think in a perfect world, perfect world, if you had the money and the infrastructure and you wanted to, you could sequence the genome of everybody every year, say, and you know, if you see warning signs that things are happening, then you could be prepared for it or act in advance of it. But I think just given the money involved, the infrastructure and things like that, you know, that sort of thing isn't going to happen anytime soon.
There's a question from Angela Sweet. Are you more likely to develop Ewing's in childhood if you've had a previous condition in very early childhood, for example, leukemia?
As to the best of my knowledge, only through if you've received treatment. So there has been some research to show that if you receive treatment earlier in your life for another cancer, then there is a very slight increased risk of bone cancers arising later on. So, for example, if you had some very strong chemo or radiotherapy, obviously the problem with radiotherapy is it doesn't just target the cancer, it targets normal cells as well. And if that radiotherapy just happens to hit TP53, or it happens to hit your chromosomes in such a way that you get that fusion gene, then that is going to increase your risk, unfortunately.
Ali Gartland has asked, is it not possible that as an individual you might have your own drivers and that there aren't common drivers?
That may well be the case. This is something that it's just one of them other complications, isn't it? You think you've cracked something, but instead of answering one question, you've just asked another 10. So it may well be that you have your own individual drivers, but as far as I'm aware from the research that's been done, particularly for osteosarcoma, the majority of patients show the same abnormality.
Okay. I've got one more question, but just before I move to that question, one of the good things about us being able to do these webinars is that people can access them from wherever they are. So I just wanted to give a shout out to Janet Panak. I think that's right. She's in Indiana, in the USA. So I'm not sure what time it is there, Janet, but thank you for joining us. Okay. Another question that was asked in advance is from Nicole Croft. She says, our 6-year-old son has metastatic Ewing's and we are near end of chemo and radiotherapy treatment. Is there anything upcoming he may benefit from or any advice that can help with recurrence? It is something obviously really worries us and we would like to make sure we know everything we can to help. I'm going to throw that question actually open to you, Darrell, but then to Zoe and Vicky as well, if you want to unmute yourselves.
So what I'd say in the first instance is, as their son is currently going through treatment, it's difficult to say what the outcome of that is going to be. But so what's likely going to happen is that their son will receive a post-treatment biopsy where they'll find out how effective that treatment was. Now, unfortunately for bone cancer, it is a very crude and subjective test because a histopathologist will look at the tumour cells under the microscope and they pretty much have to guess what percentage of that tumour has been killed by the chemo and the radiation. And there's this figure that flies around, 95% necrosis. So that really is the first indication of how things have gone and how things are going to go. So if you get a really good necrosis rate, you know that the tumour is responding well to treatment, then that's good news. But what I would say is to prepare, because you don't want to get into a position later on down the line where you wish you'd have done something to save some time beforehand. So what I would say is that even though their son is going through treatment now, I would look for a clinical trial, and we can help them do that via BCRT. We can look for a clinical trial that's happening now, so that we don't want the worst to happen, but if it was to happen, we've already set up in place the mechanism to get their son onto a clinical trial, because then that is going to be the next best thing. But obviously that takes time. It takes time for the oncologist to liaise with the clinical trial team, to then register with the trial. I think something that is going to happen for bone cancer, and what is going to have to make its way through to guidelines, is that at the end of treatment, patients aren't just left and then followed up with x-rays. I think what needs to be part of routine practice is that clinical trials are found and ready, and patients are almost pre-registered to go onto these clinical trials if the worst was to happen. So you save a huge amount of time going from end of treatment to the clinical trial. So for me personally, that would be my first step before I even found out about how well the treatment has gone. I would have something in place, ready. So as I said, that's something that we could probably help them with, and speak to their oncologist to set up as well with the trial manager.
Vicky or Zoe, would you add anything to that? You're just on mute, so hold on, I've unmuted you both.
Yeah, there's not really much to add. So yeah, please do get in touch if you've got any questions around clinical trials, and we can contact some of the clinicians that we work closely with. We can get a list together of ongoing clinical trials and some advice on the types of questions to ask, because lots of nitty-gritties with clinical trials of eligibility criteria, but we can give you some pointers to ask.
Well, okay. There's another question that's coming in. It's just being written, but I also have a response actually. So Angela, who asked about leukemia, if you've had leukemia before, could it lead to Ewing's? So she's just responding saying, so the risk is from radiotherapy, not from genetic changes from blood cancer. So no link between the two.
I believe so, yes. So I think that the Ewing sarcoma is a very specific genetic mutation that is linked to Ewing specifically. I haven't seen that mutation in leukemia before. Although leukemia has some of the same type of mutation where you get those huge chunks of chromosomes moved about where they shouldn't have been, that is similar to Ewing sarcoma, but the actual genes and the actual chromosomes are very different. So I think the risk would come from the radiotherapy, not the genetic change, I think.
Okay, so a question from Maggie. You said clearly that how cells escape from the primary tumour to form metastasis in the lung is a very complicated process. In your mouse model, do you know whether your synthetic RNA is making less lung metastasis because the primary tumour is smaller?
So we deliberately left the engineered tumours, we deliberately left them much longer than the control tumours, so that they got to the same size before we did our tests. And we still don't see, even in comparable tumour sizes, we don't see that spread to the lung. I didn't really go into too much detail on that slide, but we think that gene is recruiting those M2 macrophages. It's switching those macrophages from M1 to M2, and those M2 macrophages are either carrying the cancer cells to the lung, or because we've got other experiments that show that there's crosstalk at the immune tumor border. So whether these macrophages are quite literally eating away at the cancer tumor and creating a path to the local blood vessel, because we saw other genes that were involved in that process were active too. You know, that just adds to the complexity. But yeah, as an answer to that question, we left the tumors deliberately long enough, and we know that the longer you leave a tumor, then the more likely you are to get metastasis. So as a double, you know, to be scientifically correct, we were working with same size tumors between control and the silenced RNA. But even the length of time, you know, if you leave that silenced, that engineered version, you still don't get that metastasis, which was really a huge finding for us.
Brilliant. Okay, everyone. So we've been on an hour now. I've gone through all the questions that have been put forward. So thank you to everyone who has asked a question. Our next webinar is next Friday at the same time, 4 o'clock, with Professor Alison Gartland from Sheffield. If you have any questions after this webinar that you think of, or Professor Gartland, you can send them via email back to the email address where you got your invitation from prior to this webinar. If you think of any other questions for Darrell, then again, you can send them to us on email, and we'll make sure that we get responses to you. Thank you ever so much for joining us from across the world. It's really great. Just one thing just to mention, that if anybody on this webinar is a patient, former patient, or a family member who would like to take part in a Zoom-based support call, we do have those on a Wednesday, so it's our virtual cuppa. And if you're interested in that, just email us at support@bcrt.org.uk. But for now, thank you ever so much, everyone, and thank you ever so much to our guest, Dr. Green. You were very informative, really good presentation. Thank you. Right. Bye, everyone.
Thank you.